Hello students, let us study application of biology in industry. In this case, we are going to study enzyme production and application. Enzyme production is the synthesis of enzymes through biological, chemical, and biotechnological fermentation processes from the available feedstocks such as corn staple liquid or molasses. Enzymes are proteins that catalyze specific biological reactions, and these are widely used in industries such as pharmaceuticals, food, textile, and biofuel industries. When we see microbial enzyme production, microorganisms like bacteria, fungi, and yeast are the primary sources of industrial enzymes. They can be genetically engineered to overproduce such specific enzymes. Aspergillus from fungi and Bacillus from bacteria are the two of most applicable organisms used to produce enzymes. Why these groups of microorganisms are so important? Because they are non-pathogenic and do not produce antibiotics. Let us see stepwisely how we can produce enzymes in factory. The first thing we need to do is selection of microorganisms. In this case, we have to choose a microorganism that is capable of producing the desired enzymes. The second step is fermentation. That means growing microorganisms in a controlled environment with nutrients like carbon and nitrogen sources. Third, optimization. We need to optimize parameters like pH, temperature, aeration, and agitation for maximum enzyme yield because microorganisms in this instance require optimum or constant conditions. Fourth, harvesting. After fermentation, the enzyme is extracted from the culture medium or biomass. And lastly, purification. The enzyme is purified using filtration, centrifugation, and precipitation mechanisms. In the case of harvesting, if the enzymes are extracellular enzyme, the liquid feedstock is filtered from the organism and the enzyme is extracted from the liquid feedstock. If the enzymes are intracellular, the microorganisms have to be filtered from the feedstock and these microorganisms are then crushed to release enzymes that were inside the cytoplasm and the enzymes are then extracted with water and other solvents using filtration, centrifugation, and precipitation techniques. Let us see some commercial uses of enzymes. Proteases in washing powder used for dissolving stains from egg, milk, and blood, removing hair from animal hides, in cheese manufacturing process, and in tenderizing of meat. Lipase act as flavor enhancers in cheese production and in washing powders for removal of fatty stains. Pectinase has been used in clarification of fruit juices and in maximizing of juice extraction. Amylase has been applied in production of glucose from starch. Let us see biological washing powder. Biological washing powder is a type of laundry detergent that combines enzymes that are designed to break down stains, dirt, and green from clothes. Majority of commercial enzyme production involves protein digesting enzymes, that is protease, and fat digesting enzymes, that is lipase, for use in the food and textile industries. When enzymes combined with washing powders, they are effective in removing stains in clothes caused by proteins like blood, egg, and gravy, and fats, example grease, that are large and insoluble. So these enzymes that's combined with washing powders digest protein and fat to produce small soluble molecules which can pass out of the clothes. So biological washing powders save energy because they can be used to wash clothes at low temperature and without boiling water. However, If the enzyme are put in a water at high temperature, the enzyme become denatured and lose their effectiveness.